The overall goal of this protocol is to purify and isolate primary porcine brain endothelial cells in order to set up a tight in vitro blood-brain barrier model.
This method can help answer key questions in the blood-brain barrier field, such as drug delivery, permeability, receptor trafficking, as well as the tightness and polarity of the BB. The main advantage of this model is the high yield of primary brain endothelial cells that can be used to create an in vitro blood-brain barrier model which has high TIR and low permeability.
To begin this procedure, place the brains in a sterile flow bench and gently wash them with 1 liter of pBECs in a beaker placed on ice. Next, carefully remove the meninges from each brain using fine-tip forceps. Using a scalpel, scrape off gray matter from the brains one at a time and transfer the isolated material to petri dishes containing 20 milliliters of DMEM F12 placed on ice. For initial fragmentation, run the gray matter material through a 50 milliliter syringe without the needle. Repeat the procedure for all brains and pool the collected gray matter material together. Transfer the isolated gray matter material with DMEM F12 medium to the grinder tube of a handheld tissue homogenizer. Homogenize the material by making 8 up and down strokes with a loose pestle, followed by 8 up and down strokes with a tight pestle. Next, isolate the capillaries by filtering the homogenate using a 500 milliliter blue cap bottle with filter hole.